For millennia, humans have been fascinated by their own bodies, how our organs are formed, how they work, what causes them to become diseased. Over the past few years, our knowledge has taken big steps forward thanks to organoid biology. That's what we'll focus on during this episode of The Pursuit of Precision, the science advancing individualized medicine. I'm Kathy Wurzer. Organoids are tiny 3D tissue cultures that are derived from stem cells. According to science.org, researchers have created a veritable zoo of organoids, including livers, pancreases, stomachs, hearts, and kidneys. These are like mini organs, which gives researchers new insights into human development and disease, and it could revolutionize fields like drug discovery. Joining us right now is Dr. Mitchell Lazar. He's the director of the Penn Diabetes Research Center at the University of Pennsylvania. He's also the Willard and Rhoda Ware Professor in Diabetes and Metabolic Diseases at the University of Pennsylvania. Good to have you here, Doctor.
Nice to be here, Kathy.
Also with us is Dr. Tomas Kozicz with the Mayo Clinic's Department of Clinical Genomics. He's also with the Department of Laboratory Medicine and Pathology. He's a professor of Biochemistry and Molecular Biology. Doctor, good to have you here.
Good to have you. Thank you.
Dr. Kozicz, I'm going to start with you. All organoids begin as stem cells, right? What happens after that?
So basically, what happens that these stem cells, we can use different factors, and then with these factors, you can unlock genetic codes of different organs, and they start to develop into specific cell types, or even if you let them, in three-dimensional organoid structures. And as you said in your intro, now we have really a wide variety of organs we can develop from liver to brain. And not just that, but even within the brain, very specific areas of the brain can be developed, like midbrain or cortical areas. So I think this is really something revolutionary. And I have to say that I might be biased because I love this system. And I think this is really a great promise for future, not just, therapy development, but also understanding the disease. So as I said, I might be biased with the organoids, but I love them, and also I love the potential that this technology can give us in the coming years.
Dr. Lazar, I'm thinking because organoids can model the liver and the stomach and the pancreas and the like, it can reveal genetic differences that affect the organ's function, obviously, but may it also give us a roadmap as to how the body might respond to specific drugs?
Well, absolutely, Kathy. Organoids have advantages over mouse systems, for example, or rodent systems, because it's the human genes that are involved in the cells that are in these organoids. And many drugs, their function requires or works on aspects of the genome, which are very different between humans and other species. So we can use the organoids to measure, for example, in liver, metabolic parameters that drugs might be used to treat diabetes or to treat lipid disorders, sort of the good things that drugs do. And we can also look at the bad things that drugs do, some of the side effects that are caused by deleterious actions on the liver or on, for example, fat tissue and other tissue that we study in this way.
Mm-hmm. What are the advantages? And maybe both of you can speak to this. What are the advantages compared to current methods of using organoids when it comes to developing medications or learning about their actions?
I would say that, while animal models are very useful, there are many differences between the structure and functions of the cells that compose an organ or an organoid and the interactions between the cells that comprise the organoid. And these differences include not only the genes themselves in the genome, but also the proteins encoded by those genes that then in turn compose the cells and organs. So human organoids allow scientists to study the human aspect of the system, as well as differences from one person to another.
Dr. Kozicz?
Yeah, I fully agree with that. So as Dr. Lazar said, it's not just, the genetic background that differs, in between humans and for example, rodents like mouse and rat, and that is very hard to model in mouse models. And I think that has a very significant limitation also when we try to understand the disease, but especially when we develop treatments. And I think in this regard, organoids and also iPSC-based, so induced pluripotent stem cell-based model systems could be a fantastic tool, really bridging this gap between, the human and the animal research at this point.
Dr. Lazar, I want to kind of drill down a little bit on your research. You have worked with both adipose and hepatocyte organoids. Can you give us examples as to how these might have been used to support drug development or getting the right drug to the right patient?
Absolutely. So as I mentioned, we can take a drug and we can expose the organoid to the drug, and we can measure good things and bad things that the drug might do. And one thing that we've done is, with the understanding that not all people respond the same to drugs, we've hypothesized that one of the differences might be the differences in the genome of the people. So our genomes are composed of billions of bases, which sometimes are used alphabetically. We use letters to describe them, A, C, G, and T. And even though we have billions that are the same, there's about 5 million that are different between two unrelated individuals. And we believe that some of these differences can be used to predict, because they're causal in some of the differences, again, in the good and bad things that drugs might do. So by taking stem cells from, let's say, 10 different people and differentiating them into making liver or hepatocyte organoids out of them and then exposing them to certain drugs, we can see if there's differences that are reproducible from one person to another, and then see if those can predict the actual side effects or benefits of the medicines in people.
Wow, interesting. So when you first started to work with this, what were your expectations?
Well, we kind of went after what I would call low-hanging fruit, in the sense that some of the drugs that are used in the clinic actually work on proteins that work by actually reading the letter code of the DNA. And one of those classes of drugs are glucocorticoids, drugs like prednisone or hydrocortisone, that are used to treat all sorts of inflammatory diseases, ranging from rheumatoid arthritis to inflammatory bowel disease to lupus, asthma, and so on. And they are terrific, and millions of people have been exposed to these drugs. But unfortunately, with chronic use, people develop side effects that are metabolic, that are effects due to effects on not immune cells, which are the good things, but effects on the liver or on fat cells. And so what we did was to look at liver organoids, hepatocyte organoids, and ask about metabolic effects of glucocorticoids on each of, in this case, 11 different people. And we found examples of single bases that affected how the drug worked in the genome that we thought could possibly predict which people, if you knew that they had this code in their body, would be either protected or, unfortunately, deleteriously affected by getting glucocorticoid drugs like prednisone and getting diabetes, for example. And so that's what we did. We did it all in the organoids because it's safe and it's not in people. And then we went back to studies where people had been studied prospectively and found that we could indeed predict, from what we found in the organoids, who among the people that were treated with glucocorticoids would have an increase in their blood sugar or diabetes.
Now, I want to bring Dr. Kozicz into the conversation because you study patients with mitochondrial disease. And I'm wondering, how do you use organoids in your research?
So when we started to work with these stem cell-based model systems, I got really very excited because, the specific type of mitochondrial disease that I'm studying, that is related to the genetic material which is in the mitochondria. As you may know, that every human cell has two genetic materials. One is in the nucleus and one set is in the mitochondria. And unfortunately, at this point, now it is possible, but up to 2 years ago, it was not really possible to edit the mitochondrial DNA. And that resulted in really a serious limitation in any model systems that we could develop. And specifically, we had no animal models and not even other cellular models, which were edited regarding the mitochondrial DNA. The only tool we had that was maybe patient-derived fibroblasts or maybe muscle cells. But of course, again, they do not really represent the metabolism and also the proteome of, let's say, of a brain or a cardiac cell or a muscle cell. And then I got, contact with a colleague who started to work with organoids and iPSC-based model systems. And we developed these first human and specifically for this mitochondrial disorder based models, with neurons and also brain organoids. And we started to study them. And I was really, how would I say, positively surprised how wonderful data we could really get out of it. And then after that, when we correlate these, with known physiological or pathophysiological changes related to a specific disease presentation, we could really understand, what's going on. And we were able to mechanistically study and also find, ways to modify these responses and then develop or actually discover novel therapies in these mitochondrial disorders.
All this sounds fascinating, but I'm wondering about the limitations of organoids. What have you both found? Dr. Lazar?
That's a great question. And I would say that the main limitations have to do with the fact that it's not currently possible to exactly replicate the organ that exists in the body. We'd love to make a liver organoid. We sometimes call it a liver organoid, but livers have lots of specialized cell types, not just the hepatocyte, which is the main liver cell. And those cells have to be in a certain relationship to one another. And furthermore, when we study that organoid in the lab, it's not in the context of a normal blood supply or blood system where they could communicate with other tissues via hormones. They don't have a nervous system to, supply them with neural signals. And this will be the challenge, to figure out a way in the lab to replicate or simulate the really physiological situation for an organ like the liver.
I agree, and we have the same limitations if you think of the brain, also the heart. So we can develop certain cell types, and then it becomes more complex, But of course, it is far from the complexity of the human brain or from the human heart itself, and especially with regard to the brain, And I think also in the heart, if you develop these organoids or these cell models, they are actually in a very early developmental stage. So we cannot really follow up on them when they fully develop. And that is also an advantage because we can recapitulate the human development, in these systems, and then can give us answers, how the brain develops in these early stages. But of course, at one point it stops because at this point of technology, we cannot really resolve the issue of the blood circulation, innervation of these organs, and that limits how they further develop. And I think we have to be aware of these limitations when we interpret, the mechanistic studies and also drug discovery studies. But I think the field is developing so fast, and now we have, people who put together different organoids, like brain and muscle, and they start to innovate and they start to develop, connections. I also know colleagues, they develop these 3D matrices, and then within those will these organoids develop, and then it allows some sort of circulation. So I think we will see a fantastic improvement in these technologies, which will somewhat, decrease these limitations in the future.
This may be a very simplistic question, and I apologize, but I wonder, the models would not include an immune system, right? Which would likely influences how disease develops. So that would be, I think, also a challenge.
Yes, I agree.
Yeah, agree.
Okay, so just wondering about that. Also, I would think another hurdle would be they remain hard to churn out in large, consistent batches needed for drug screening, right? And other efforts.
Yeah, I think that there's always going to be biological variability in some of these. I completely agree, though, with Dr. Kozicz and his optimism about the future. And, you could even imagine, it's not even imagination, it's happening, 3D printer kind of things where you could sort of type of cell. You'd almost have a color by number system where you'd have a compartment you want your immune cells, a compartment you want your hepatocytes, a compartment you want your nerve cells, and you would just kind of print it out. So future is rosy.
What will be the biggest potential of organoids for precision medicine, do you think, when it comes to at the bedside for patients?
So if I may, from again, maybe this is a biased sort of approach. As we said, these organoids, they really represent the human genome, the human proteome with the limitations that we mentioned. And I think that's not always true when you have a look at, or actually study a mouse system. So I think in drug development, this is also going to be very important that now with these systems and these models, we can really study disease-relevant tissues. So we can actually look at disease or actually treatments, or drug discovery or repurposing drugs using disease-relevant tissues, let it be brain or heart or muscle. Like in mitochondrial disorders, as I said, so far, if there were animal models that was possible to do some drug discovery, or they used yeast or maybe some fibroblast models. But a yeast and a fibroblast model is not really a brain. It's not really a heart. It's not really a muscle tissue or a muscle cell. So I think the organoids and these cell-based models, human cell-based models, really give this opportunity for us. And maybe one more thing, and I think that Dr. Lazar is also maybe looking in this direction, what could be a fantastic personalized or precision medicine in this field, that we know that some drugs, certain disorders, they respond to multiple drugs, and one individual responds to one drug and the other individual responds to another drug. And we don't know that at this point, how this response is going to be when we start treating these patients. And I think as this technology develops, just imagine how the genomic technology develops, and now it's basically almost an everyday tool to have a gene sequence. Maybe we can also develop and really have these systems so fastly developing and also ready for use that, for example, a patient comes in, we take a fibroblast, develop a disease-relevant tissue, let it be brain or liver or a muscle. We try these different treatment options, and we can tell that which treatment is really beneficial for that individual. Maybe even we can actually design the proper dose of a drug. So I think this could be a possibility, and I really believe that this, within a decade, will be a possibility to have these sort of bedside, really precision medicine based on these organoid or human cell-based technologies.
What do you think, Dr. Lazar?
You were talking about, bedside work. Well, the amazing thing is that we can take the skin cells from an individual and we can convert them back to stem cells using just 4 factors. There was a Nobel Prize given for this technology. And by doing this, you avoid any concerns, religious or other, about the use of stem cells, because you're starting with an adult stem cell. There's no embryo involved. So you kind of go backwards, and now you have something that you can go forwards and look at, make a heart, make a liver, make a brain with the exact DNA of the person who was ill or you wanted to learn more about. So that part of this, as opposed to, let's say, having a stem cell line, which is just one person's DNA that's not completely related to your patient. So I think that's another avenue in which the sort of, if you will, bedside to bench and back is almost limitless in terms of the possibilities.
What is the most exciting part of the research you're doing right now, both of you? Because I know you're both involved in some very interesting research. What is exciting you right now, Dr. Lazar?
Well, we're a little bit related to my previous answer. We're trying to personalize the effects of the drugs and figure out which predictive bases, letters in the genome, can again predict which people get diabetes. And then we really want to test this. We've shown in some systems that we can do it, but we've kind of picked the easiest systems. For example, our best data now are in children treated with glucocorticoids, but the children didn't start with diabetes, they didn't start with a weight problem. So it's really easy to tell which ones developed it and which ones didn't. But the problem we run into with some of the patients that have lupus or rheumatoid arthritis who are adults is they may already have diabetes. They've already been treated with steroids. They already be overweight. And so it gets harder for us to tease out over time what is truly the effect of the drug versus just other circumstances. And in a lot of these type of studies, if you have, what are called false negatives or false positives, like you say a person did respond, but they really didn't, it really makes it hard to do the analysis. So we're working with clinicians to try to do a better job of that.
Dr. Kozicz, in your research right now, what's the most exciting part of it for you?
I think we have two most exciting maybe aspects. One is really the mechanistic studies, and I'm not just working in the mitochondrial disorders, but I also work together with my wife. She's a clinical geneticist, and she's focused and really specialized in congenital disorders of glycosylation. And as I said, both of these disorders share one thing. It's at this point, it's very current technological level. It's very challenging to design animal models or really have animal models which are useful. So we were really limited in our toolkit, to study these disorders. And the stem cell-based technology that gave us an enormous possibility to study the mechanism of disease, and also how the different tissues or maybe organs are affected by these disorders. And the other aspect, which is really exciting, that we use these human cell-based model systems to discover and also validate novel drug treatments for these rare metabolic disorders, which are really not having treatments at the moment. And this is really exciting. And it also uses drug repurposing, which may not be that exciting, with regard to intellectual property. But I think the drug repurposing in using these systems could really accelerate moving a drug target to a clinical treatment. And we have some examples where such a drug repurposing effort within 3-4 years actually from the bench basically now is in a clinical trial.
Gentlemen, thank you so much for the insightful conversation, and thank you for the work that you're both doing. We appreciate it.
Thank you.
Dr. Mitchell Lazar has been with us. He's the director of the Penn Diabetes Research Center at the University of Pennsylvania. Dr. Tomas Kozicz is with the Mayo Clinic's Department of Clinical Genomics and with the Laboratory Medicine and Pathology Department as well. If you have questions or comments about what you heard today, do send us an email. It is precisionpod, P-O-D, precisionpod @mayo.edu. And for goodness sakes, follow us wherever you get your podcasts. We will have future conversations about a number of different topics in precision medicine. I'm Kathy Werzer. Until next time, here's to your health and well-being.